Hello, my name is Parag Chitnis. I'm one of the principal investigators in the biomedical imaging lab at George Mason University. Today, I wanted to share with you some research opportunities in our lab for prospective PhD master's students and postdoctoral fellows. So, the three active projects that I wanted to discuss are ultrasound augmented wound healing, photoacoustic sensing of bioelectric activity, and AI enhanced photoacoustic tomography. So, in the first project, we are working in a consortium of universities and industry partners led by Columbia University to develop new methods for improving healing outcomes for recalcitrant wounds. Being ultrasound experts, our role is to help develop active bandages with integrated ultrasound transducers, which can enable imaging of the wound bed, and they can do so in 2 dimensions and 3 dimensions. We're also working with partners at MIT and Columbia to incorporate implantable devices made from hydrogels that can serve as tissue scaffolds or drug delivery beads, and these can be activated using ultrasound. So the ultrasound transducers in the bandage can provide images like these or focus ultrasound in this fashion to activate these implanted devices in a wireless manner. And this stems from our prior work at Columbia University, where we demonstrated that many hydrogels can be made ultrasonically responsive. For instance, if you're using NIPAM hydrogel devices like shown here, NIPAM is thermally sensitive. So if you heat it, it contracts. So think of it like a sponge that's loaded with drug. And if you squeeze the sponge, the drug can come out. And that squeezing action is induced by heating the hydrogel in this case. So what we're doing is using focused ultrasound, or FUS, to actually achieve this heating in a wireless and non-invasive fashion. But what's also very nice about these hydrogels is that the contraction is nicely dependent on the temperature. And even more importantly, these thermally induced contraction is reversible. So when you heat it, it contracts, and you let it cool, it resumes the original dimensions and shape. So this is a very nice platform then for us to deliver therapeutics locally by controlling these devices using ultrasound. And we've already demonstrated the efficacy and concept of this type of approach in simple animal models. Here we show the implanted device prior to actuation. This is immediately after actuation. And this is showing the release of fluorescent compound with the device removed. So we can see that this does work. So now we're going to leverage this approach to improve wound healing outcomes. The second project I wanted to discuss is photoacoustic sensing of bioelectric activity. And this particular project is motivated by the fact that there is a continuing need to understand cell signaling in the brain to better understand brain function and diseases. Now, in vitro or superficial cortical layers of the brain, it's not too difficult to do optical fluorescence methods and understand these input-output relations, neurons, or connectivity in large networks of neurons. But to do these types of studies in larger length scales in deep tissue or an intact mammalian brain is very difficult with fluorescence methods. So we think that photoacoustic approach might benefit here. Now, photoacoustic is a hybrid technique which combines optics and ultrasound. So in traditional ultrasound imaging, sound waves go in the tissue and the echoes come back out. In photoacoustic imaging, light goes in the tissue, which gets absorbed by the local chromophores. And that absorption produces sound waves, which can then come out. And we can make an image with that in the same way as we would with ultrasound. But now the information we get is molecular and functional. So it's different from ultrasound. And then to do this, to use photoacoustics for imaging brain function, we are focused on these three aims. So the first being developing novel voltage sensing reporters for photoacoustic imaging. Second is to non-invasively deliver these in the mouse brain. And then third is to develop instrumentation for photoacoustic imaging of activity, functional activity in awake, behaving, and moving rodent models. So currently I have a PhD student who is focused on this first part, and we are using some very interesting new DNA-based nanostructures in collaboration with Professor Venziano at Mason. And this has been very exciting new front for us. And we have lots of interesting results here as well. Here's an example. If we have a cell culture with a fluorescence setup and a patch clamp, patch clamp is basically a microelectrode shown here in this image that can manipulate the cell membrane voltage of just this one cell. And if we track the fluorescence signal of this cell, the background, and the control cell, which is not patched, we can see what would happen when voltage of this cell is changed. So if we apply an input voltage voltage function here on this cell, we can see indeed that the fluorescence signal changes according to that step function, but the control and the background, they do not. So right now we're actively seeking students who would be interested in taking this work further to explore new nanoconstructs and dyes, to take the work in vivo with animal models and develop instrumentation for imaging mouse, and to perform neuroscience and neurological studies. The third opportunity is somewhat related to the second one, which is to develop computational and deep learning methods to enhance photoacoustic tomography. So when we want to image intact brains or do deep tissue imaging, we have several challenges. And to overcome some of these imaging challenges, we resort to tomographic approaches where we have large number of sensors detecting the signals from tissue. But the reconstruction of the image is then an inverse problem. So if we have an object like this that we're imaging, and we're using a large number of sensors that surround the object here, then the photoacoustic waves will propagate from the object. We detect it from the sensors, and here's our sensor data. And we can use this sensor data to reconstruct an image with methods, for example, time reversal reconstruction, and we get an image like that. So this is fine in concept, but in practical implementation, there are several challenges. So while in optimal scenario, we would want an infinitely large number of sensors surrounding our object that would give us a reasonable image, in reality, we don't have those options. Sometimes we can only place sensors on one side of the object. And if we do that, the reconstructed image will not be complete. Or it would be very difficult to have a large number of sensors because we then require a large number of electronics channels to go with that. And that also would cause a degradation of image quality. In fact, reality is probably a combination of both of these situations, which is even worse, and then we would not get nearly as good of an image as the idealized scenario. So we are investigating if deep learning approaches such as convolutional neural networks can be used to compensate for these degradation in image quality. So right now we're focused on imaging the mouse brain. So we are developing new types of convolutional neural networks that are optimized for imaging the mouse brain. And we train them on these types of vessel images right now, and then test them on mouse brain images shown here on the right. And we're already getting some very exciting good results here. In recent publication, we showed that our approach to use deep learning to directly go from a raw data to image performs comparable to actually better than the image to image deep learning approach, and far superior than the conventional approach of like time reversal. And this is the ground truth. So these are the three different projects where we have opportunities for graduate students and postdoctoral fellows. If you want to chat about these opportunities or read papers, feel free to reach out to me. I can share papers and discuss these research projects with you in more detail. Some information about our PhD program and admission requirements can be found here at our department website, and our lab website link is shown right here as well. So we do hope that you reach out and join us at Biomedical Imaging Lab at George Mason University.